Good morning, everyone, and welcome to the next in our series of HIRN webinars, the Human Islet Research Network. I'm Joyce Nyland. I'm the PI of the Coordinating Center for HIRN, which we call the HIREC, Human Islet Research Enhancement Center. Today's topic is an introduction to a program called the Human Pancreas Analysis Program, or HPAP. And we're going to give you some real-world examples of successful data analyses using this program and its database. Our presenters today will be Dr. Klaus Kaestner from the University of Pennsylvania, Dr. Maria Hatzoglou from Case Western Reserve University, Dr. Golnaz Vahedi from University of Pennsylvania, and Dr. Paul Wang from University of Pennsylvania as well. So as an overview for today's webinar, first, I'll give you a little bit of background on HIRN if you're not familiar. And then for our presenters, which now are freezing. Okay, hold on. Okay, never had this before. But our presenters are going to give you a couple of background also, Dr. Kaestner, on HPAP. And then some real-world examples, Maria and Golnaz, of some recently published work that they've done. Then Paul is going to tell you how you can access the system and the database, which is called PancDB. Then we'll hold the questions and discussion for the end. We'll have Q&A. And if you could please use the, under the reactions, raise your hand if you would like to ask a question at the end. And we'll call on you and try to get through as many as we can. Now, I don't know why my screen is frozen. So I'm going to maybe have to stop sharing and try again. Sorry about that. Okay. Still is frozen. Layla, perhaps, oh, there we go. Okay, now here we go. So those are the presentations. And then, as I mentioned, we'll have the Q&A at the end. So the HIRN is a network of 5 different consortia, and there are many benefits to this type of network and structure. We can share discoveries early, develop new technologies together. We've initiated new investigator and emerging leader awards for people in the field, and we can share across the whole diabetes and islet community as we're doing today. So we really enjoy sharing what this group is developing with all of you, whether you're in the HIRN network or not. And that will be what we'll do today, is kind of show you these resources we're developing and how you can access them as well. So the network began in 2014. And here's a diagram of the 5 consortia. There's the, on the left, beta cell death and survival, the consortium on modeling autoimmune interactions, the consortium on human islet biomimetics, the consortium on targeting and regeneration. In the middle, there's our coordinating center hub. And today we're going to be focusing at the top there on HPAC, the Human Pancreas Analysis Consortium, and their PancDB database. The network now consists of over 200 investigators, 80 institutions, and it's across 9 countries. There's our URL for our HIRNnetwork.org website, and here's one of the screenshots for what's called the resource browser, which is an important resource that any of you can go to. And I hope that you will, and our speakers are going to point to you to the resources that they've developed in this browser and how you can get to them. And we now have over 1,200 entries in this resource browser, from antibodies to mouse and cell lines, viruses, vectors, data sets, and protocols. So I hope you will take advantage of that. So that's my overview. And now I'm going to stop sharing and introduce Dr. Klaus Kaestner, if he would take over this screen. He is the Thomas and Evelyn Sweur Butterworth Professor in Genetics at the University of Pennsylvania. And he's also the co-PI of the Human Pancreas Analysis Program that looks at T1D and T2D pancreata. He's going to give you an overview of this program. Klaus?
Thank you so much. Let's see if I can manage sharing my screen. Hang on, share screen. Okay, so the Human Pancreas Analysis Program is in fact 2 programs at the same time. And is aimed at both the better understanding of type 1 diabetes and also type 2 diabetes. And it's abbreviated as HPAP, and this is our logo. Give you a brief history
Klaus?
of how. Yes, sir. Yes.
Maybe you could put it in the screen share, the slideshow mode.
Slideshow mode. Yes, ma'am.
That would be a
That
little
better?
easier. Better, thank you.
Okay, good. All right. So the HPAP type 1 diabetes program was initially funded for 3 years because the NIH was worried or concerned whether such a program could in fact work. In other words, could scientists from multiple institutions work together and actually effectively obtain organs from relevant organ donors, these being type 1 diabetics, people with autoantibodies, and of course, age-matched controls. Because our first 3 years were successful, we were just recently refunded for an additional 4 years in the so-called production phase. So that was wonderful news. Also, because of the initial success of the HPAP T1D program, NIDDK decided that it would be wise to expand this program also for the study of type 2 diabetes. So now we've been collecting a great number of organ donors which have type 2 diabetes. So, well, how far are we? As of to date, we have collected 104 well clinically phenotyped and detailed analysis organ donors, 104, out of which 58 have no history of diabetes, 24 have type 1 diabetes, and 22 have type 2 diabetes. I'll give you a bit more information on these. As you can see, if you look at this is the age distribution now from newborn to about 65 years of age. And as expected, the type 1 diabetic organ donors are on average much younger than those with type 2 diabetes. Fortunately, we selected control donors, so people with no history of diabetes, to span the entire age range so that if somebody wants to do a study on type 1 diabetes, they can use age-matched controls from here. Likewise, if somebody is analyzing a type 2 diabetic individuals, they can use controls starting from here to over here. So in both cases, there's a nice set of age-matched controls available. But what do we actually do? The HPAP works together with NPAT. So these are Mingda Wang, Mark Atkinson, Al Powers, and Ali Naji. They work together with all the organ procurement organizations in the country to antibody screening and then identify those organ donors that should be selected for a study. And now the critical part is that we do as many experiments as possible on these organs. So for instance, we collect the pancreas from tail, body, and head of the pancreas tissue in multiple different modalities, so different assays can be done. And then pancreatic islets are isolated, and many experiments are done on these tissues. For instance, we have tissue analysis, not just H&E, but imaging mass cytometry to be able to profile 34 different proteins at the same time. We do extensive molecular profiling, including single-cell RNA-seq, single-cell ATAC-seq, bulk ATAC-seq, bulk RNA-seq, and also whole genome bisulfite sequencing. We do very careful islet physiology, not just hormone secretion, but also calcium imaging and mitochondrial bioenergetics. As I mentioned, we do multiplex imaging. This is imaging mass cytometry as well as codex. And we have a large team of immunologists to study the immune system as it is right near the pancreas, as in the pancreatic lymph nodes in great detail. And I should say, importantly, all data are immediately deposited into our database called PancDB, which you'll hear more about at the end. I should say a condition of accepting the award from NIH was that all data are immediately deposited into the database, not after publication, but immediately after data generation. Okay. And I'm going to give you also some evidence of the HPAP impact, some of the critical papers that were produced by HPAP over the last 4 years. For instance, we developed single-cell mass cytometry for the analysis of the endocrine pancreas. We then also did tissue multiplexed imaging mass spectrometry to look at tissue sections in great detail. Oops, whatever. We did a great study on single-cell transcriptomics of human islets from very young individuals to old ones and compared them to type 2 diabetics. We published an important paper in Cell disputing a recent study that suggested that there's a specific TCR, BCR dual expressing cell that is relevant in type 1 diabetes, and we found this is not the case. We have a great study from Golnaz, I'll speak in a few minutes, about how there's genetic variation, that how this actually impacts 3D chromatin conformation in T cells and thereby alters gene expression. And an overview of all of HPAP was published in Diabetes a couple of years back by the leaders of our group. And with that, I'm done, and I turn it over to Maria. Thank you.
Great, thank you, Klaus. So Maria is Professor of Genetics and Genome Sciences at Case Western Reserve University. And if she would begin sharing her screen, I'd be happy to hear her experiences in using PancDB. Can you get it into the single slide mode? We're seeing the notes as well.
Am I ready?
Yes, but you need to put it in single slide mode. It's side by side right now.
Oh, I see. Just 1 second. So this place. And I'm okay.
There, good. Thank you.
So I would like to thank you all for the opportunity to present at the webinar. And what I will tell you today is adaptation to chronic ear stress enforces pancreatic beta cell plasticity and how we think it is lost in T1D. So we adapt in environmental insults. And what we know is extinction is the result when a species fails to adapt to a changing environment. We therefore ask the question, is T1D development a failure to stress adaptation? Stress adaptation was described over 20 years ago as 3 stages: the alarm stage, the resistance stage, and the exhaustion stage. We ask the question, is T1D development a failure to stress adaptation, considering the higher stress that beta cells are exposed to? So do we have exhaustion of adaptation? To study this, we have to think how beta cells respond to ear stress. We know that proinsulin synthesis has an inherent component of ear stress in the beta cells all the time. However, additional factors, microenvironment, metabolic alterations, genetic mutations, therapeutic drugs, increase the ear stress on the beta cells. Beta cells adapt by reprogramming their transcriptome and translation in a way to downregulate things that overload unfolded proteins in the ER and increase the ability of the ER to process these processes. In addition, we should consider the ear stress not as a continuous stimuli, but rather stress, adaptation, and recovery cycles. So is T1D developing because we fail to reproduce the cycles or because the adaptation is disrupted? All cells respond to ear stress by decreasing global protein synthesis and increasing selective mRNA translation for proteins which are important for the adaptation of the ER under ear stress conditions. The data that I will show you will support a model that says that if I have episodes of ear stress, that they are far apart, I have beta cell plasticity, and cells can lose and return to their beta cell function, maturity, and mass. However, if I have too frequent episodes of ear stress, then the cells do not return back, the cycles are disrupted, and this may be the reason that we have T1D development, beta cell loss, and failure. So to study at the molecular level, we used Min6 mouse beta cells As an ER stressor, CPA, which is reversible, we can wash it out. And the physiological stressors, of course, will be nutrients, cytokines, mutations, etc. But this is our model. And we ask the question, how do cells respond to stress and how they recover? It is shown here at the left that when MIN6 cells are exposed to CPA, the stress markers accumulate as expected for the integrated stress response, which is shown on the right. At the same time, protein synthesis dramatically is inhibited at 1 hour and returns partially over the time of adaptation to chronic ER stress. This partial recovery of protein synthesis is very important, not to be full recovery, because full recovery will increase the ER protein synthesis, and this will increase the load of protein synthesized in the ER and therefore the collapse of the beta cells. So, is it reversible? Yes. In panel B, you're going to see that after 18 hours of stress, if I remove the stressor, all the stress markers are gone. Panel C, protein synthesis recovers, and panel D, survival of the cell returns. Therefore, I can have with CPA reversible cycles of ER stress. What happens to proinsulin? Very interesting pattern for proinsulin levels. At CPA 1 hour, which is the acute response, the levels decrease, stay low for a few hours, and return back to even higher than the control levels. Upon recovery from stress and wash out, the levels are maintained, and they gradually decrease. Interestingly, the subcellular localization of proinsulin is in cytoplasmic inclusions at CPA 18 hours, which are disappearing when the stressor is washed out. This suggests that proinsulin may accumulate perhaps in premature granules. So the fate of this proinsulin can be very interesting and can explain the proinsulin which is found in T1D. In the plasma. We were interested to identify molecular markers for the adaptation to chronic ER stress of the MIN6 cells. And to do this, we used ribosome profiling that maps changes in the transcriptome, therefore mRNA abundance, or translatome, therefore ability of the mRNAs to be translated, that we call translation efficiency. And we use 3 stages: the alarm stage, using CPA treatment for 1 hour, the resistant stage, CPA 18 hours, and repeated cycles of CPA exposure that I will not show you the data. So after we obtained all the data, we asked the question, how many genes are regulated early, late, up early, up late, etc.? With a very important group, the chronic down, because we're interested to know what the cell attenuates in order to protect ER function. And as you can see, the chronic down group has a predominant pathway shown in pink, beta cell related genes, insulin secretion, and a few more. So this told us that beta cells downregulate beta cell function genes in order to survive ER stress. Then the next question, and here we can see a validation of this with a few genes. Notice GLUT2 gene decreases in panel C and is restored during washout, and MAF-A decreases in mRNA levels and restored, and same thing for the protein in panel D. In contrast, in MIN6 cells, as you can see in panel B, you find that at 1 hour of stress, there is no change of these genes. Only at 18 hours of stress, which is the chronic adaptation, you see the decrease of these beta cell marker genes. So when the cell then decreases certain gene expression, What does it increase? Because whatever it increases will be to protect the ER. Whatever it decreases is also to protect the ER. So let's see then what it increases. To do this, we compare MIN6 cells to a very simple cell line, not specialized, mouse embryonic fibroblast cells, MEFs. And you see here in color, 1,534 mRNAs are upregulated in MIN6 cells in mRNA abundance, when 567 in MEFs. When we look at the pathway of these two groups, the enrichment pathway is protein processing in the ER. This pathway has 150 genes involved. 68 genes were observed for the MIN6 cells and 38 for the protein processing in the ER. We can add in the 68 10 more genes, which were found to be induced at the level of translation, as it is shown at the left panel. So the difference is 78/38. So already the beta cell is trying very hard to increase expression of additional genes to help the ER protein processing. What are these genes? The 41 genes indicated here, which are different, are ER to Golgi transport, vesicle transport, COP2 coated vesicles. So an extra effort is being put forward by the beta cell to really process ER synthesized proteins. So the next question was, what happens in disease development, T1D or T2D? In order to address this question, we thought it's the best to use genes which are regulated in beta cells, but are not regulated in MEFs. So we can find the kind of the beta cell specific regulome. So these were 334 genes here, which are shown that some of them are increasing in abundance, some of them are decreasing in abundance in the middle panel. The pathway analysis was dominated by the beta cell specific function. We took these 334 genes and we looked in control and T1D islets and in control T1D from the HPAP program, single cell analysis. And the focus was the beta cells. So in the islets, we identified 11 genes, almost all of them related with beta cell function, being downregulated, as it's shown in panel C. And in the beta cells, shown in green, a lot more genes were downregulated. And over here, you can see that we have in beta cells 73 genes, when in the islets 11, and then we took the 6 that they are common between the islets and the beta cells, and we're looking in patients that they have T1D for less than 3 years or up to 7 years. And as you can see, the level of expression of these 6 marker genes is decreasing. So I told you before that the MIN6 cells make a big effort to really upregulate ER protein processing. So we asked the question, what happens to these 78 genes that they are involved in ER protein processing? We looked at the beta cell, single beta cell analysis, the data from HPAP, and we found all of them are downregulated except BIP. This is in contrast with the MIN6 cells that they show upregulation during the adaptation phase to ER stress. So we can conclude from this that the data are consistent with T1D diabetes having a failure of adaptation and a decrease of beta cell function genes. To better understand the relationship of the different populations of beta cells with the UPR marker genes, we used a machine learning approach to determine the distance of gene expression of the UPR signature genes, the GO pathway is here for you if you are interested, plus the beta cell identity genes, over the beta cell populations, healthy and disease together. So let's look at the first panel. The first panel shows that the healthy individuals have heterogeneous populations of cells in 3 different populations, where development of disease is more the upper populations of cells. They cluster for the UPR markers, as you can see here. If we look at expression of the insulin gene, the bottom group has a higher level of expression, when BIP, who is more a sensor of UPR, of stress, of the UPR, is more with the disease development. When WFS1, which is an adaptive gene, it is increasing during adaptation to chronic ear stress, is more with the healthy individuals. So this put forward the hypothesis that healthy cells have heterogeneous populations, and maybe these heterogeneous populations really show that the beta cells undergo cycles of stress, recovery, and return, and this is what we are seeing in these 3 different populations of healthy cells. And when this is lost, the recovery, then the cells progress to this different state that they cannot be beta cells anymore. So this is, we try to show this here at the left. Maybe we have different phases, active, resting, and recovery, that they perfectly go from one to another as the cells are exposed to stress, and they return back to homeostasis. And disorder develops in diabetes when this cycle breaks. So let's take then the healthy cells at the right and look at different markers. Notice that the bottom population of cells expresses insulin, MAF-A, NKX 6.1, and PDX1, or beta cell specific gene expression. However, NKX22 and PAC6 also is expressed in an additional population. Perhaps this shows the return of the cells back to the bottom group. Maybe it's the closing of the cycle, since these markers come later to stop any other gene expression, alpha cell, etc. BIP is expressed to the upper population, which will suggest it is the less beta cell-like population of genes, of cells. So we put forward this model. Beta cells exist in their physiological environment in different states as they undergo cycles of stress, recovery, and back to homeostasis. And this can perfectly keep everything in balance. However, when disease develops, too frequent cycles of stress or too high intensity of stress, this balance is disrupted, and I cannot have any more return of beta cells to their mature phenotype and in a functional form. So what I told you then today is maybe T1D develops because the cells lose adaptive plasticity in response to chronic ear stress. And the data say that gene expression signatures of cells from T1D patients indicate the loss of cell identity and suggest lack of adaptive plasticity. So this work was done by a very talented research scientist in my group, Wen Chen, and it couldn't be done without the help of Klaus Kaestner, who helped us with Long for the data analysis. And it was in equal contribution with the lab of ESA Engine at the University of Wisconsin-Madison. And of course, it couldn't be done without the PancDB data that even as we were able to access and use them. And I encourage you, it's very friendly site to really obtain the data and analyze your own data. Thank you.
Great, thank you very much, Maria. That was great. So now we're going to hear another example from Golnaz as to how she utilized the PancDB data. Golnaz is an associate professor of genetics at the University of Pennsylvania. Golnaz.
Good afternoon, everyone. It's a wonderful pleasure to talk to you about HPAP data use case presentation, especially after the very exciting presentation from Maria. as a specific example from the what we heard from Maria about ER stress, a more general question which many of our are interested is that what are the transcriptional differences or perturbations that happen in the islets or in the pancreas during type 1 diabetes? A particular assay that has gained a lot of interest over the last few years because of the technological advances is the single-cell RNA-seq profiling. And as Klaus very nicely described in the beginning, the islets have been isolated and the single-cell RNA-seq profiling has been performed across all the multiple donors that have been collected from the beginning in the Kaestner laboratories. And what I'd like to talk to you today is, as I think in the beginning, Klaus mentioned that these immediately as every data set is generated is deposited on PancDB. But at the end, we really are interested in a collection of how together multiple donors in different groups are able to transcriptional profiles of donors across different groups, and data integration can tell us about common cellular pathways that are disrupted in type 1D. So extra efforts is really required to combine donors at various points of times and really extract the common perturbed pathways. So I'm going to talk to you about our kind of phase 1, combining 24 different donors collected by HPAP in terms of transcriptional profiling perturbed in type 1D. So, but my today goal is primarily I will focus on an effort that has been kind of spearheaded by the UPenn HPAP investigators, but I also want to give you provide to you two other cases and hopefully encourage you that the HPAP single-cell RNA dataseq and many other datasets generated by HPAP are easily available and can be integrated in multiple manuscripts and really help us understand what is disturbed and disrupted in type 1D and type 2D. So the first case, which is on bioRxiv, and you can see the manuscript, is really a kind of outcome of our powers combined, as Klaus described in the beginning, of procurement initiated by Ali, single-cell profiling performed by Klaus's lab, and computational efforts led by Babak Faryabi, a colleague at Penn, and also two really fantastic postdoctoral fellow, Gregory Schwartz and Maria Fesolino in my lab. And of course, we are really fortunate to have collaborators across HPAP group to be able to kind of expand these efforts across many other areas. So this effort of combining and integrating the donors as in kind of the first one, which we have kind of about 70,000 cells equally distributed across donors. And for those of you working on single-cell profiling data sets, in addition to really getting into the cleaning of the data, removing doublets and those type of pre-processing efforts, there are two major computational tasks that need to be done. First, the cells need to be integrated and has to be clustered based on their similarities. And then the cells have to be labeled based on what they are and how they can match to the legacy knowledge of what we know about various cell types in different tissues, in this context, the pancreas. So for cellular clustering, we use this computational technique that was developed in Babak Faryabi's lab called Too Many Cells, which not only clusters cells, but also is really powerful in visualizing cells that are clustered together in terms of their similarities of transcriptional profile, but you can immediately see the cellular relationship. So you can really see how if the cells were clustered as alpha cell and beta cell, they have more transcriptional similarity between these cells compared to ductal cells. So visual representation of similarity and relationship is something that was very important and was kind of a primary choice for us to use this particular clustering technique. So we have performed these clustering analyses and annotations. The data is available, and I will talk to you how you can access to that and ask really specific questions that you're interested in. In the remaining few minutes, I just want to talk to you about one particular novel biological observation we had from the analysis of these data sets. Surprisingly, we found that in these islet preps, again, that part is not surprising. In these islet preps, because of the impurities, there's always a percentage of ductal and acinar cells that are present. And fortunately for us, we were able to identify the ductal cells in type 1D donors had really interesting features. In particular, we were first noticed that the ductal cells in type 1D donors from the single-cell RNA-seq data that is unbiased, measures all the genes, that the ductal cells express MHC class II proteins, which is usually a cell surface marker for antigen-presenting cells such as dendritic cells. But quantitatively, we were able to find these cells across all donors, but find their increased frequency or presence in type 1D individuals. So of course, single-cell RNA-seq measures RNA and has its own limitations. We wondered whether MHC class II proteins are expressed on the surface as a protein level. In the CyTOF data that was also generated by HPAP and is also accessible on PancDB, we are able to really, again, quantitate across donors the higher percentage of the HLA-DR expressing ductal cells or MHC class II positive as ductal cells. In addition, the imaging mass cytometry data, again, generated in the Kaestner lab and deposited on HPAP as every one of these donors being available, revealed quantitatively, and as you see here as a specific example, the ductal cells expressing KRT and the co-expression of HLA-DR and KRT suggesting, and again, elevated frequency and number of these type of cells in type 1D donors. And the fourth method that we really want to get a little bit in higher resolution, which is lower throughput, was the immunohistochemistry and really being able to see the co-expression of these ductal cells, the ductal cells expressing HLA-DR by the co-expression of these two markers. So all of these four data modalities now undoubtedly show that the presence of these cells in type 1D and indicate that the exocrine compartment has some unique features in the type 1D donors. In most recent data that we have that I probably don't have time to talk to you today is we are really struck by finding that actually the type 1D cells in type 1D donors really have a lot of resemblance to the dendritic cells themselves. In other words, they seem to act as some sort of tolerogenic dendritic cells by suppressing, potentially, this is our speculation, we need more experiments and studies to really validate this. But it seems that these ductal cells acting or feeling or looking like dendritic cells have features that resemble the tolerogenic dendritic cells, which their job is to suppress T cell function. In other aspects of this study, again, I didn't have time to show you, we found a really interesting resemblance in gene expression profile of many different cell types in autoantibody positive donors and type 1D donors, and the ability to rank transcriptional profiles with the GAD level is something that we have recently found, and we are really excited in being able to link directly transcriptional modifications across the genome to the actual autoantibody level and really linking that to specific cell types and cell signatures across the pancreas. Again, these were some of the really exciting questions that the reviewers of our manuscript asked us. If any of the people in the audience are those reviewers, I really appreciate that because they were really constructive and may help us really get into more in relating the autoantibody positive donors to type 1D donors. So it takes a village to really get these huge efforts done. I want to thank Klaus, Kaestner, and Ali Naji, and of course the first authors of the manuscript. Again, you can access to the bioRxiv paper here. In the remaining few minutes, I want to talk to you about 2 other cases of being able to use the HPAP data sets across these donors, the same donors that I actually showed you in the manuscript that is currently under review and is available as is preprint. So in the number 2 case, this was a integration of the same data set in a report, in a study that was published in Nature Medicine by actually one of our colleagues, Dr. Dan Rader at Penn. This study was a really giant amount of 10,000 plus whole genome sequencing linked to electronic health record data in Penn Medicine Biobank. And what this effort led the investigators to a list of genes that they knew there are some mutations associated with different kind of genes and different kind of traits, including type 1 diabetes. And they really didn't know what to do with those lists of genes. And in particular, we were able to help them by looking at one of the genes that they found, RGS12, which they found a loss of function mutation in this particular gene, and by integrating and looking at that particular gene in our data set, we found that the expression of macrophages in islets of type 1D donors was significantly lower compared to controls, suggesting that maybe some of the donors that we had or some of the shared mechanisms that is involved in type 1D has to do with loss of function of this particular gene. The third case study of utilizing the HPAP data relates to a really wonderful study that was published by Al Power's group. And in this publication, which has been cited 40 times since only December of 2020, Dr. Power's group showed how examined very carefully the expression of ACE2 and TMPRSS2 in pancreatic islets and showed that these proteins are actually not expressed despite what the field or some literature indicated in beta cells. And we were able to include a large number of cells from HPAP integrated with multiple other studies, validating this particular observation or kind of enhancing the validity of many data sets and many other experimental modalities that has also shown this. So as Joyce mentioned, the resource browser is really where we provided a web portal for being able to type in the gene of your interest and get to exact same level of knowledge that we are able to get within the HPAP investigators, and we would be extremely happy if you and your team are able to use this. And please reach us, ask us if there is any way we can help with access to these data sets and these web portals. Another place we have deposited our data is the Cell by Gene website from the Chan Zuckerberg initiative. What is really interesting about the single cell genomics field is that the SORAT package has really kind of become the standard way of data analysis. So in this particular submission, you can easily download the SORAT object, and you can skip months of analysis and pain that we have gone through to really get to the level of cells, donors, and you will be able to really answer a lot of different questions at the more for more computationally savvy individuals. And also there is a browser that you can look at and explore the genes based and looking at the UMAPs and cells as they are annotated. So with that, I want to thank my lab and again collaborators Klaus, Ali, and Babak, and would I think be happy to end here. Thanks.
Great, thank you, Golnaz. Very interesting talk. That's terrific. and appreciate all the great examples of tying back into the HPAP and PancDB. And now Dr. Wang, Paul Wang is going to tell you about how you could access PancDB. Dr. Wang is the Senior Project Director at the University of Pennsylvania Institute for Biomedical Informatics. Paul?
Thank you. So you've heard great things about HPAP, and you may wonder, how can I get data for my own research? So I'm going to talk about the data downloading from PancDB, the HPAP database. So this is a website for PancDB and its homepage. You can pretty much just go there and browse and explore and see if we have the data you need. It's a long website. I know it's hard to memorize. So what you can do is to Google using the key term HPAP or PancDB. The first hit is PancDB. It's just that easy. And then if you're new to PancDB, I will suggest you start with the data status page, which provides you an overview of all the PancDB data. So it has two parts. One is the demographics and the clinical overview with a summary of all the data we have. And the second part, experimental data, it pretty much looks like this, a plot. And the title provides a list of all the assays we have in PancDB. And then in the first column, it gives you a list of all the donors. So just visualizing this plot, it can give you a quick idea of the availability of specific assay data that you might be interested in. So after this data status page, you might want to look into the donor information. So this is a donor page. The left part is a filtering panel that allows you to search using specific criteria. And the right panel is the donor page that provides basic clinical data, clinical information. And then in this page, you can download data for a specific donor and view the HLA information. Or if you want to know more information about a specific donor, you can just click this button, view the HPAP donor ID. So for example, you want to look into more information for a specific donor, then you will see this donor case page with all the clinical data for this specific donor and also all the other data related to this donor. So it also provides you the image visualization function and access to other assay data. So on this page, you can also download all the clinical information for all the donors by clicking these two buttons. And on this donor, you can just browse through, going to the next donor, or choose download full data for this donor, and then you will go to the data download page. What I need to mention is you can definitely go to this page directly if you are sure, if you know what data you want to download. And this page similarly has a filtering panel for you to search. And also the right part is a file explorer type of file selection tool that allow you to select the files you want to download. After you finish selecting, you can just click the button, download selected, and then it will download your selected files on the web. And you can also download metadata here, or to view the data structure of the PancDB using this button. So we realize you can easily see that the size of all the data for a specific donor can be really large. It's not possible for you to download all data you need on the web. Then we provide FTP downloading function. So in this page, it provides all the detailed information for you to follow, so you can log into our FTP server, search your data, and even run command line to download data easily. So if you have any questions, need help, feel free to use this contact form or shoot us an email directly using this email address. We'll be here, we'll be glad to help you with any questions you have about PancDB. So this is a list of all the work groups for the PancDB development. Thank you.
Excellent. Thank you for that overview, Paul. And that's great timing. Appreciate all of your talks. They were very excellent. Now we'll see if there are any questions. If you would like to ask a question, please go to reactions and hold up your hand, and we can call on you and take you off mute and put you on camera and see what there is to ask. So Paul, maybe, well, we're seeing if there's any question. Oh, here's one. We'll go down here first. Franzi Boot, you want to unmute and ask your question? Or is that, no, that's maybe my hand. Never mind. Paul, do you want to maybe mention the integration that's about to happen with IIDP as something that's a coming feature between HPAP and PancDB and IIDP?
Yes, I didn't mention is we have collaborations with other HIRN groups for integration. For example, the image file, we have a collaboration with Pancreas. So you can navigate between PancDB and Pancreas easily. You can just jump between the two websites for image file visualization. And Joyce just mentioned is another collaboration that's under development, that is the clinical data search and the downloading for PancDB data on IIDP website. So what we're really close to finish this task. That means you can search the PancDB clinical data on IIDP website, and if you want to know more detailed information about a specific donor, you can easily jump back to PancDB website for more information and more data types.
Great. Yeah, we're looking forward to that collaboration being in place. Should be within a month or two. Good. Did anybody have any other questions, either on the talk content or about PancDB access?
I have a question. Can you hear me?
Yes, Vincenzo, go ahead. Thank you.
Yes. Hi, Klaus, and congrats for the great sessions. I have a question regarding the so many tools that are developing for single-cell RNA-seq when it comes to figuring out what is happening to a certain population with respect to another. So depending on the algorithm you are using, you can have impressively different results, even though overall the clustering of different cell types may remain the same. Have you tested the ability of some of the tools that you are using to crosstalk, let's say, with Monocle 3, which seem to be another tool that people are using as well?
Yeah, I think this might be a question that Golnaz can answer best, as she's worked a lot on different analysis pipelines for this.
Okay, thank you.
Sure, yeah, that's actually one of the, I think the field of single cell evolved so quickly that it's been wonderful for us as investigators, but also at the same time really overwhelming because there are so many techniques and each of them optimize one aspect of the analysis and compromise some others. So in our particular case, we actually compared clustering and annotations across too many cells and SORAT. And we also used Monocle for pseudotime analysis, etc. So I think the one particular strategy that we also have is we think if a biological observation is reproduced by multiple different computational techniques, is something that we really like to follow up and continue its validations and the follow-up mechanistic investigation. So if something is real, has to happen across more than one computational technique. So that's something that we actually do very rigorously, and that's why it takes time to really come up with final annotation of the cells and clustering that is reliable for differential expression analysis, etc.
Thank you.
Okay, thank you. Any other questions for any of the speakers?
Maria, I have a question for you regarding the frequency of ER stress. Beta cells see so much stress all the time. How do you describe or identify these episodes?
So the way I envision it, and again, I'm envisioning this, right? The way I envision this is like proinsulin synthesis. It cycles, right? We have glucose-stimulated secretion, more insulin comes in, stores. The same way that we have proinsulin synthesis, if we have a diabetic person or we have cytokines around, or if we have an inflammatory response, maybe this proinsulin synthesis is much higher. And this what is enough to really push the cells to that window. And continuous exposure to cytokines can do it too. In other words, I'm dreaming now to really add cytokines in the same condition and see what it will do, if it will accelerate the loss during cycles. That's how I'm looking at this. But one thing I really would like investigators to think about is it's impossible a beta cell to withstand continuous stress. It has to go in recovery. Impossible. Like in tissue culture, they cannot stand it. So it has to be a mechanism of cycling of the cells of resting periods.
Thank you, Maria.
Thank you. Any last question before we sign off for today?
I have one for Maria.
Okay.
Yes.
You touched something, a topic that is really interesting when it comes to ER stress. So by the very term stress carries a negative connotation. But during development, ER stress below certain threshold is also a good thing. Cells undergo a cycle in and out of some level of ER stress all the time, especially during development. How do you set some standards, some threshold not to be passed, over which the process will become irreversible?
So if you are a secretory cell, right, if you secrete, you are in big trouble. So during development, if you have ER stress, you will strengthen the ER function. And this may be needed for development. If you are a secretory cell that has been differentiated, then you will be in big trouble, right? Because now, in order to survive, you have to take away what it makes you the specific cell type. That's what I try to say, that the ER stress is there to help development, to help survival, to even promote survival better than normal, in normal cells in some cases. But the problem will come when you are a secretory cell, liver cell, plasma cell, beta cell. So if we look in development, what secretory pathways we have, and if they relate to ER stress in non-secretory cells, it will be interesting.
All right, thank you.
Great. Thank you, Maria. Okay, I want to thank all of our speakers, Klaus, Golnaz, Maria, Paul. We appreciate it very much, and we hope that you will go to the HIRN resource browser and avail yourselves of those resources and go to PancDB and download and explore the information. Okay, thank you very much. I think we'll end for today.
Everyone.
Thank you.
Bye.
Bye-bye.